Director of the CDC Office of Genomics and Precision Public Health. I would like to welcome you all to our monthly webinar. We are in for a treat today. Before I introduce our speakers, just a couple of notes about this monthly webinar series. It has really changed in character and landscape over the last couple of years during the pandemic. We have strived to do monthly events at the intersection between research, policy, and practice. And our intent, primarily from the CDC perspective, is to try to identify genomic applications that are ready for prime time, inform the various stakeholders and the research community, and try to integrate them into clinical practice with the idea of improving population health. So if you go to our website, we've had a number of different topics over the last couple of years that range from polygenic risk scores to heart disease and cancer and so on and so forth. We have two upcoming webinars, one next week on October 6th on host genomics and COVID-19, where we are with the science and its implications for clinical and public health practice. Be sure to register and tune in. And later on in October 24, we have a webinar on the intersection between coronary heart disease, family history, and elevated lipoprotein A. Be sure to tune in to this as well. So today we are in for a treat. This has been a very rich season for us because we are from our office calling for all public health scientists to pay attention to this through the eyes and ears of the All of Us research program. And I don't want to take too much time out of our speakers this morning, but suffice it to say that we at CDC have had a very keen interest in the successful implementation of the All of Us research program at NIH, because the data that will be coming out of it would be really a boon for both clinical research as well as public health practice. And over the last few months, we've developed a data use agreement with the All of Us research program that allows all CDC scientists to access and register to analyze the All of Us research program data through the cloud platform. So our objective this morning is fairly simple. We would like to review with you the All of Us research program, how it's designed and the current status of it, discuss its potential for providing important population data to inform both disease prevention and treatment activities, as well as tackling health disparities. And then last but not least, provide some kind of a hands-on training session for scientists who are interested in accessing and analyzing All of Us data. So our speakers this morning, and I'll introduce them in succession, Jeff Ginsburg, who is the Chief Medical and Scientific Officer for the All of Us research program at NIH. He leads the Division of Medical and Scientific Research and responsible for helping set the scientific vision and strategy for the program. Jeff is well known to many of us. As a matter of fact, Jeff, you were a speaker not too long ago, but you were still at Duke University. That was maybe early last year, where he was a professor of medicine and pioneered translational genomics activities within the program there. The second speaker will be Sherry Shully, who's the Deputy Chief Medical Officer from the All of Us research program. In this role, she helps set the scientific vision and strategy and oversees the establishment of ancillary studies for the All of Us program. Before joining All of Us, she was the team lead and senior advisor for disease prevention at the NIH Office of Disease Prevention. And before then, she was at NCI, where I met Sherry many years ago. She was in the Division of Cancer Control and Population Sciences, where we teamed up on many public health genomics activities, including our last 4th National Conference on Genomics and Public Health. Last but not least, Kelsey Mayo. Dr. Mayo is a scientific product and portfolio manager at Vanderbilt University Medical Center. She's a multidisciplinary scientist with many years of precision medicine research experience and the founding member of the All of Us Data and Research Center. Dr. Mayo recently led the successful development and launch of the Research Workbench and the All of Us Research Program cloud-based analysis platform. She began her career at the Beckman Scholar as the Georgia Institute for Technology, and later earned her doctorate in designing advanced therapeutic as an NSF Research Fellow at Vanderbilt University. So I think a few words before I turn it over to Dr. Ginsberg. Want to remind everyone we are recording these talks. Hopefully this whole webinar will be posted on the All of Us YouTube platform. After this, everybody is muted, but we will open the chat box in a little while, maybe in the next 5 to 10 minutes, so that you can start adding your comments and questions throughout the presentation. You can ask questions, make any comments. This should be hopefully an interactive session as much as possible. So without further ado, I'm turning the mic to Jeff to lead us off in the discussion. So Jeff, thank you for joining us.
Thanks a lot, Muin. It's really great to be here. Before we begin, I just want to say our thoughts are with our colleagues and their families who live in Florida with Hurricane Ian and the devastation that we're seeing happening there, hoping everybody is safe. As Muin said, I'm Jeff Ginsberg. I'm the Chief Medical and Science Officer for the All of Us Research Program. I've been in this role for 10 months and it's been terrific, but I really think, particularly with the engagement of the scientists on this call, the best is really yet to come. We're really excited about expanding our work with the CDC and CDC affiliated researchers to the All of Us Research Workbench. And as the CDC is the nation's health protection agency, we hope that the All of Us Research Program can be a really significant resource for better understanding health and disease, and that will ultimately have an impact on our nation's health. As Muin indicated, the All of Us Research Program, and you're going to hear more about this from Sherry, really has a commitment to serve those who have been underrepresented in biomedical research. And we believe that population of participants in a longitudinal population study of this type is really unprecedented. We hope that today's discussion will catalyze new epidemiology, not just genomics, although I know that's a favorite area for Muin and his team, but really to instigate and catalyze other approaches to disease understanding and trajectories using new epidemiologic approaches, and to give us a better understanding of individual and population health. So with that, I'd like to turn this the podium over to Sherry Shelley, who, as you know, is the Deputy Chief Medical and Science Officer of the All of Us Research Program, and who's been with the program nearly since its inception and has been an invaluable member of our scientific team. Sherry?
Thanks so much, Jeff. I'm trying to share my PowerPoint, and I just want to make sure that it's coming up.
Not yet.
Okay. I don't know if I need some technical assistance, Anya.
Did you press on share screen?
I did, yes.
Okay, no, that's not good. I can go ahead and share it from here if you want me to.
Let me try this.
Never happened before.
Yeah, go ahead and since I sent this slide to you, if you don't mind just sharing from your end,
yeah.
Great. Sorry about that. Always have to have a backup just in case. So thank you so much, Muin and Jeff. Really excited to be here. And I'm going to give sort of a background on All of Us and sort of where we are as a program and which data we're collecting. And Kelsey's really going to get into the nitty gritty of how our researcher workbench works. And we really hope that this is an interactive session. Next slide, please. So first of all, what is the All of Us Research Program? You may have heard of it before, but we're going to dive a little deeper. Next slide. So we are one of the largest, richest, most diverse biomedical data sets of its kind. As Jeff mentioned, we really focus on underserved populations or communities that are underrepresented in biomedical research. We hope to have 1 million or more people from across the United States that really reflect the broad diversity of the United States. So currently, we have data available from over 370,000 participants. 80% of those are what we call underrepresented in biomedical research, with 45% being of racial and ethnic minorities. Next slide, please. Next slide, please. So our mission is to accelerate health research and medical breakthroughs by enabling individualized prevention, treatment, and care for all of us. And we do this in 3 ways. We nurture relationships with 1 million or more participant partners from all walks of life for decades. We're going to deliver one of the largest, or have started to deliver the largest, richest biomedical data set ever, making it easy, safe, and open to use as possible. And you'll hear a lot about this from Kelsey. But just because we have created this really robust data set doesn't mean people will necessarily just come. So we hope to catalyze a robust ecosystem of researchers and funders who are happy to use and support it so that it really can be the resource we hope it will be for decades. Next slide, please. We enable research discoveries that drive more precise approaches to care. And so we've been engaging with people in communities who have typically been left behind by medical research. We are really combining biological factors, including genomics, as well as social determinants on a large, inclusive scale. We're going to be following participants as they move, age, and grow for decades. And we hope to make this as easily accessible to any researcher with a secure internet connection and a data use agreement, which Muin already mentioned CDC does have one. So hopefully the folks on this call will be able to figure out how to get on after Kelsey and I finish here. Next slide, please. And we are collecting several different data types. So currently we have surveys from over 370,000 participants. These include lifestyle, access to care, medical history, both family and personal, as well as different surveys that include social determinants of health and experience during the COVID-19 pandemic. Many more surveys will be coming over time, but we currently have 7 in the mix. We also link to electronic health records, and over 258,000 of our participants actually have linkages to electronic health records, and their data is available within the researcher workbench. So we can really get some outcomes and have some hard clinical data to link to. We collect physical measurements, including blood pressure, heart rate, BMI, as well as waist and hip circumference from our participants. We've been linking to wearables. So currently we link to Fitbit data. And so basically we have physical activity and heart rate data from participants who have been able to link their Fitbits to their All of Us account. And currently we have over 100,000 whole genome sequences and about 170,000 arrays on our participants currently available in the researcher workbench, with many more to come. And then we have a huge biobank that we are storing biosamples, both blood and urine, as well as saliva from some of our participants. And those will be available for ancillary studies down the road. Next slide, please. I mentioned already that the first batch of genomic data was released in March. This is already the largest whole genome database out there. We have uncovered almost 600,000 unique variants within our diverse cohort. And if you can look here on the right-hand side of the page, as you start to pair different data types together, you can see that you can really create a robust data cohort based on what's already publicly available within our researcher workbench, which Kelsey will dive into. I also want to mention that we have added genomic analysis tools, including HALE and Plink. And so you can actually get in there and utilize the data that we have. Next slide, please. So I mentioned already that we are trying to make all this data accessible to researchers across stages and settings. We have over 3,000 registered researchers from 400 institutions across the country. We are actually have over 2,400 active projects ongoing, and you can actually see the list of research projects as well as publications on our researchallofus.org site. And we are also linking to what are the conditions that are being studied. Next slide, please. We also want to make sure that in addition to building a diverse participant cohort, that we are creating a diverse researcher cohort. So we are making sure that we are promoting responsible and ethical use of data, return of value to participant community, and really accelerating our research impact. We are trying to encourage students and early stage investigators to come to our resource and use it, as well as access to researchers from various institutions, including minority serving institutions and historically Black colleges and universities. Next slide, please. Kelsey will certainly get into this, into the weeds, but we have both a public data browser as well as a behind the scenes, get your hands dirty in the data. But this is an example of our public side of things that sort of, you can look at aggregated overviews and interactive previews available to everybody. So if you wanted to get in and see what was what we currently have in our database before actually registering to become a user of the Researcher Workbench, our researchallofus.org page really has some nice data snapshots and really talks about what different data types we have and how many people. Next slide, please. I've already mentioned, next slide, that we currently have over 2,400 active projects within our research projects directory, which is really great. We are trying to link them as much as possible by the condition that's being studied. We also hope that this directory will help to forge collaborations. So if you see a project on there that looks like something that is in your wheelhouse or that you study, you can actually reach out and hopefully form a collaboration, as well as seeing what's being done. This is also useful for our participants because they can really see what type of research is happening in real time with the data that they graciously provided to our program. Next slide. So Kelsey will get into the how to actually create an All of Us user account. I put the QR code here just to make it easier. And so, so hopefully folks can log on pretty quickly, but Kelsey will walk through it in more detail. Next slide. And then of course, we have several communication tools, including our Researcher Roundup, that will talk about the latest news and insights coming from All of Us. So if you'd like to hear more about that, you can certainly register to get that Research Roundup. Next slide. So next slide, please. It certainly would not be possible without lots of really great partners and community providers, as well as our participants who provide the data for us. So this is as of August 2022. And I feel like every time I show this slide, it gets, the icons get smaller and smaller because we have to fit more and more on the page. It's the only way that we can actually get this done. And next slide, please. And here's our consortium members, which are over 300 across the United States. So with that, I'm actually going to turn over to Dr. Mayo, who will give us an interactive preview of our Researcher Workbench.
Thank you so much, Sherry. A quick check, is everybody able to see my screen? So we are able to see the homepage of the All of Us Research Hub, is that correct?
Yes,
Yes.
that's correct.
Fantastic. Well, thank you so much, Sherry and Jeff and Nguyen, for this wonderful overview. I'm so excited to be here today. As everybody mentioned previously, my name is Kelsey Mayo, and I am here representing my team at the All of Us Data and Research Center. So when you think about this incredible program that really is a huge effort of all kinds of partners all across the country, where my organization comes in is we really are responsible for taking the data that, as Sherry noted, our participants have so graciously provided, storing this data very securely, organizing it, and ultimately making it available on our research hub here, so that the broader scientific community is able to really drive forward discovery. So my goal today is really to give you this live demonstration of our research hub, really point out some of those tools that Sherry mentioned on our public website that are valuable for researchers interested in learning more about the data we have available currently, the tools that we have available currently, and certainly how to register and become an All of Us researcher. I'm then going to spend the second half of my demonstration in our secure research enclave. So the part of the research hub, which allows you to access and analyze row-level data as long as you are a registered and approved All of Us researcher. So with that being said, let's go ahead and get started. Here on the screen, you can see our homepage of this Research Hub public website. So the URL, if you'd like to follow along with me, is researchallofus.org. And this hub is really trying to connect the broader research community with more information about the program, more information about the data. So what data is All of Us collecting? One of the things that we've really been proud of recently is we've just in the past month or so surpassed our 500,000 participant milestone, which is a huge accomplishment, and kudos to many of our enrollment partners all across the country. And certainly we do, as a result of the hard work of the All of Us Consortium, have a wealth of data here available for research. So this is a place where you can start to really learn about that data. It's also a place where you can start to get familiar with what we mean by the researcher workbench. And we're going to get a jump start on that here today in our webinar, because you're going to get sort of a live demonstration of the researcher workbench and the tools that you would have available to do your projects. In addition, we have a whole section of our public website dedicated to discovering the work of the broader All of Us research ecosystem. So as Sherry and Jeff indicated, you can really look into things like our public website's list of publications. So this is where you can see work that's already been peer reviewed and published by the All of Us community. This is also where, and I'll show you this shortly, you can dig into our research projects directory and really read about some of the science that's ongoing. In addition, this is the place where you can start to access some really helpful support resources. I'm going to repeatedly emphasize how wonderful this part of our website is. Keep this link handy. It is very valuable. And then finally, our public website really is this front door through which you will register and apply to become a researcher. So today we're on a research team, and our goal is to see if we can improve our ability to assess risk for type 2 diabetes in diverse populations, because diversity really is a strength of this program. And so one of the things that we'd like to do together today is to conduct a genome-wide association study looking for genetic associations with type 2 diabetes using All of Us's multi-ancestry cohort. So the first question that we might have as researchers is, what phenotypic and genotypic data or genomic data are available within All of Us? And as Sherry mentioned, probably the best place to get a hands-on understanding of our data is our public data browser. So if you want to navigate to our data browser, we kind of sprinkle links throughout the website, but one way you can do this is to go to Data and Tools and click Data Browser. And so if you can imagine that you're standing above, looking down at the research program's data pipeline, the 500,000 participants who've enrolled and contributed data regularly have their data curated, and that curated data is made available for research. One of the things that we as a program value incredibly is our participants' privacy. So we do a ton of things to protect participants, one of which is we tier our curated research data. So always, we're striking this balance between how do we protect our participants, ensure their privacy as best as we can, and also maximize the scientific utility of this data to really drive research impact. One of the things that we've done is to create several tiers of data, and you'll hear me mention these throughout the presentation. A public tier, which is what we're going to see here and what powers the data browser on our public website. A registered tier and a controlled tier. These bottom two tiers of data do require that you register and be approved to access data through our researcher workbench, because this is row level participant data, even though we've taken privacy preserving measures to protect our participants. So what we're looking at now is an aggregated summary version of our research data set. And this data browser is a great place to get familiar with the size and shape of our data. So here you can see that this tool is broken into several sections. We do show you that we have EHR data coming from, for instance, conditions information from billing records and participant electronic medical records. And here you can see we have over 24,000 medical concepts on over 227,000 participants. And that's just one example. We're also getting data on drugs, labs and measures, procedural information. This all comes from electronic health records. Scrolling down a bit, you can see that we have information coming from the biospecimen in the program. So here we have, almost 100,000 participants who have had whole genome sequencing done, and over 165,000 who have some array data. In addition, some participants have been invited to contribute baseline physical measurement data. So we have over 300,000 participants that have some of these baseline physical measures. We also have our initial and growing set of digital health technologies data, and a really robust and powerful library of self-assessment data in the form of these surveys. I'm specifically going to draw your attention to our basics questionnaire, which every participant takes as part of their enrollment into the program. And this is where you have the opportunity to see this core demographic information. And over 370,000 participants have taken this survey. And these covariates are incredibly important, regardless of really what type of analysis that you're wanting to run. So today, we're diabetes researchers. Another thing that we can do in this tool is just to search across all of the data types here. And this tool is going to update to show us what might be most relevant to my search term. Here you can see that there's likely going to be some related information to diabetes, unsurprisingly, perhaps, coming from our EHR, and also from our self-assessment of personal medical history and family health history. So if we wanted to dive in a little deeper on this tool, you could click conditions here, and you can see that in order of descending participant counts, we have a little over 48,000 participants who have at least one mention of diabetes in their electronic medical records. And specifically, we have over 45,000 who have at least one mention of type 2 diabetes. If you want to get a little bit deeper here, you can. So on our public website, you're able to drill down just a hair to see some specific crosstabs. So we allow you to look at things like sex assigned at birth or age, but also, and I think this is a valuable thing, especially as you're getting familiar with our EHR data, you can drill down a bit into our source information here. So when you think about the 48,000 participants who have been coded with type 2 diabetes, or excuse me, 45,000 with type 2 diabetes, you can see what that information really encompasses. Because one of the valuable things that the program has done is we have, I think, over 40 different healthcare systems which are contributing data. And we do the work to harmonize this information to standard vocabularies, a common data model here. So you can see that we're using SNOMED, and you can see how this information is arranged and organized. As researchers, I find this pretty exciting. It seems like we have a pretty large sample size here to get started with our analysis. But since we're interested in genomics data as well, let me briefly go back to our data browser and highlight one more aspect here. If you click on this genomic variant card, not only can you see basic demographics, and now these are self-assessment, so this would be self-reported race and ethnicity information on participants with whole genome sequence or array data. You can also go through here and you can see variant search information. So I have the ability to search by a gene, a variant, or a specific genomic region to look at allele frequencies in the All of Us cohort, to look at certain population information about participants that have whole genome sequence data in All of Us. So using our example here, I want to use the example of the TCF7L2 gene, which is really commonly known to be associated with increased risk for type 2 diabetes. And specifically, let's imagine that our team is really interested in this variant, which is known to be a risk factor for type 2 diabetes. And specifically, if you have this specific mutation, you might have as much as a 2 times increased risk for type 2 diabetes. You can click down on this little caret here, and you can see a little bit more information about this variant. Specifically and importantly, you can look at the genetic ancestry of individuals in All of Us who have this specific variant. So at this point, we've been able to play around with a public data set. We've been able to get a sense of the size and the shape of this data, but we cannot yet do our analysis on this public website. We will have to apply to access this row level information and really get started with our analysis. Before we do that, one last thing, and Sherry mentioned this earlier, but I do want to talk about it before we move on to the secure analysis enclave. Is maybe we want to learn more about what other researchers are doing in this space. So perhaps we want to see, is anybody else studying type 2 diabetes in the All of Us research program? So one thing that we could do is visit this Discover tab and go to our research projects directory. This is where we can see more detailed information about projects. And the reason that we're able to do this is actually incredibly important. All of Us has changed the access model from what we typically see. Instead of provisioning access based off of an individual research project, we are provisioning access to individual researchers. So researchers essentially given a data passport. And once they have that passport, they are able to go in and create as many new projects on the tier of data that they have access to as they would like, whenever they like. They can wake up at 2 o'clock in the morning with a brilliant idea, create a workspace, pursue their project, but they must be compliant with all of our data use policies, and they must tell us transparently what they're doing with data. Those descriptions are what we see here today. And so, for instance, if we want to look at work that's ongoing in the world of diabetes research, we can see, who has mentioned the word diabetes within their the scientific question section of their project description. We see there are about 155 adults that have a mention of diabetes in their projects, and you can see there's really a breadth of different types of projects that the community is studying. Here you can just see a recent example on diabetes and depression coping behaviors. And you can dig in and see a little bit more about not only the questions being studied, but the scientific approaches, anticipated findings, and the research team who's conducting this analysis. And one thing I love about this program is just how many people that I see who are early career investigators or students that are really able to use this data. And so you can see here that part of this research team is an undergraduate student. Okay, so we've been able to together investigate a little bit more about the data using our public data browser. We've been able to explore what others in the community are doing on our research projects directory. I am not going to go into this in too much detail, but I do want to just tease a little bit the user support hub. This is an incredibly powerful resource. This is where you will go to look up things like you can search for data quality reports. This is where you can search for information, tips and tricks on using our tools. Find the calendar of our in-person office hours, our virtual office hours, excuse me. So I think this is just so valuable. And if there's one thing you take away from this presentation, it's please, join the research community. Please visit our public website and don't underestimate the value of the support hub. Okay, finally, we're really interested in getting on to the researcher workbench today. So hopefully you can see here that it's as simple as clicking this registration link. And right now, we have about a 6-step process, as Sherry mentioned, if you are a researcher affiliated with the CDC. Your institution has already signed the necessary data use and registration agreement for researchers, affiliated researchers to access either controlled tier data where genomic information lives, or registered tier data as well. And so you're essentially going to be able to start more or less at step 3, where you create your Workbench account. We verify your identity using login.gov, ask all researchers to complete the necessary ethics training, and then finally ask you to sign a data user code of conduct. And this process can take as little as a couple of hours. Most researchers complete this process in less than a day. Okay, so we've completed our tour of the public website. Let's spend the next part of our talk talking about once we have access. So once you have access, you will be logging in to the Researcher Workbench, which is our cloud-based analysis platform, to do your studies. Today, I've already got my account going. I'm going to just switch tabs over so that you can see my homepage here. This is the Researcher Workbench. And I want to orient you to a few key facts about the Researcher Workbench. The fundamental thing is that the unit of analysis, the basic building block of the Workbench, is called a workspace. So every time I want to conduct a project within all of us, using all of us data, I am going to create a workspace to conduct that project. These are collaborative, so I can share them with colleagues in my institution, outside of my institution. As long as they are also approved, all of us researchers, they can conduct this study with you. So in this example, you can see, and we'll go through this in a moment, that I have created a workspace for our type 2 diabetes study. But let's say that you're an investigator who really wants to refine our electronic phenotyping algorithm so that we can really have a very accurate way of identifying people likely to have type 2 diabetes within all of us. So maybe these were your workspaces that you created, and you shared them with me as a collaborator and gave me read access so that I can see the work that you're doing. Of course, you know, that's sort of the fundamentals. And one of the important things about the Workbench is this collaboration. But the other thing that I really want to highlight here is that bringing you into the cloud allows us to bring our support resources to you. So another wonderful part of this website is sort of in this hamburger menu at the top left-hand corner. Not only can you see your workspaces, the projects you're working on, but you can see an entire library of end-to-end demonstrations and tutorials, really showcasing how to get started with a number of different types of projects and data types. You can see another link to that support hub that I keep mentioning. And at the end of the day, if you need help or if you have feedback for us, please contact us. You know, we have a trained team of experts, many of whom are on the call today, who are always fantastic and willing to help. Okay, so now that we've oriented ourselves to the Workbench just a little bit, let's imagine first that let's say we're onboarding, we're getting to know this environment. One thing that we want to do is perhaps check out these featured workspaces. So I mentioned previously that we had this entire library of pre-populated code, example analyses. So we have tutorials on how to do things like get started working with the data. Since we're interested in doing a genomic analysis, we may want to start with a few of these workspaces, how to work with all of us genomic data. We may also want to see end-to-end example analyses and look at our demonstration projects here. One thing I really want to draw your attention to is this data quality report. If you would like to know and be able to replicate the code on here is how many participants have this combination of data. Here is a breakdown of their key demographics. This is a wonderful workspace to check out. And of course, we also have a phenotype library where we have taken previously published and validated algorithms. Most of these come from the eMERGE network and implemented them in the All of Us data set. So for us today, since we're interested in studying type 2 diabetes and we want to work with genomic data, so we want to be in the controlled tier, we may want to start with this previously validated phenotype rather than having to build our own. This is already ready for us to take and get a head start on our analysis. Of course, this phenotype algorithm was originally developed by researchers at Northwestern University, and we are able to attribute this work to them and really appreciate all of the investigators who contributed this work. Okay, so let's get started on our project. Going back to the homepage of the researcher workbench, and if you want to start a new workspace, it's as simple as clicking this little blue plus sign. On this page, you're going to be asked to fill out a project description, and we ask that you do this thoughtfully because this information is going to be shared publicly. It's also going to be something that we audit for appropriate use and consistency with our data use policies. But once you fill out this description, you will be able to get started on your project. Today, I've already started a project for us, so I'm just going to quickly go back and select one of those workspaces I mentioned earlier. So here is our demonstration workspace for today, and it's time for an anatomy lesson. So workspace anatomy 101, you can see that we have 3 core parts to a workspace. We have a data tab, an analysis tab, and an about tab. And just at a high level, you can think about the data tab as the place where you could access point and click tools to slice and dice this gigantic data set to select the specific participants and data that you're interested in studying for your project. The analysis tab is where we have our analysis tools that are really the workhorse, if you will, of the workbench. Right now, we support analysis using Jupyter notebooks. Programming languages we support are R and Python, and we're actively working to expand the analysis tools that are available. And then finally, we have this About tab. So in the About tab, you can see information that we entered when we originally created our workspace. We can edit this information if our project changes over time. This is also where I would share my workspace with collaborators. So once I've shared my workspace with you as our research team, I've got it set up, we're ready to go. Let's get into the data. Going back to our data tab, one of my favorite tools, and admittedly I'm a bit biased, but is our cohort builder tool. This is a tool which allows you to arrange criteria in a Boolean logic string. So include participants who have this type of data or this type of data, exclude participants with this type of characteristic, so that you can really refine the group of participants that are relevant to your study. I've created just an example cohort here. One of the things I've said is I want to include people who have at least, I think, 2 mentions of type 2 diabetes within their conditions information coming from electronic health records. This matches what we saw on our public website earlier. I also went on to say I want to see at least 1 mention of one of the following glucose lowering medications and insulins within our electronic medical records. And then finally, I wanted to exclude participants who have type 1 diabetes from this case cohort. If I wanted to go on and refine this tool works very similarly to the data browser, in that you can not only search across all domains, but you can also search within domains to really refine your electronic phenotype. And once you're done, you would save your cohort, maybe go on to build your case cohort, or excuse me, your control set. Okay, so that's the cohort builder. Let's say we've got our type 2 diabetic cases, we've got our type 2 diabetic controls. One of the next things that we're going to need for our analysis is their demographic information, because we need these covariates in order to run our genome-wide association study. So that's where our data set builder comes in. You can see here that I've already sort of pre-built a data set which pulls demographic information on our type 2 diabetic cohort. I can refine exactly what data I want to pull in for my analysis, click Analyze, and what this tool has done for me is it has actually created a custom SQL query behind the scenes that I haven't had to create. And when I click Analyze, I'm given the option to put this code into one of my Jupyter notebooks so that I can run it and pull my participants into my analysis environment. Okay. So in this example, I've already pulled up one of my analysis notebooks. So you can see here that we are looking at a type 2 diabetes phenotype analysis. This is a Jupyter notebook I actually pulled straight from our phenotype library, and I did very minor modifications to it. So again, the algorithm that we're looking at here was originally developed by researchers at Northwestern University, and this notebook allows me to attribute their work, to describe my methods, and then go on to actually do my analysis. Skipping forward to the end here, what I've been able to do in this notebook is pull various cases of type 2 diabetes and then look at a cohort based off of key demographics, things like sex assigned at birth, race ancestry information, ethnicity information, race information, not ancestry information, ethnicity information, age distribution, etc. So I've done some basic demographic characterization. Now we're ready to run our genome-wide association study. As a next step, I'm likely going to create a file with this phenotype information, my cases, my controls, my covariants, and the tool which I want to use for my genomic analysis is called HALE. And so in this example, I'm using HALE to run my genome-wide association study. I've done this in a separate notebook. I'm not going to bore you by scrolling through my code, but we can look briefly here and we can see that I've loaded my genetic information here into HALE within a Jupyter notebook. I have loaded my phenotypic data as well, which we went through earlier. I am processing all of my genomic data, getting it ready for my analysis, and then ultimately running a genome-wide association study. So in this example, I did do this. I did it on a tiny slice of chromosome 10. And one of the things that is nice to see here is we do, in fact, replicate known associations. So this is the exact variant that we were looking at on our public website earlier in the demonstration. So at this point, you have seen us work together. We went on the public website. We were able to play around in powerful tools to learn about the data, to learn about the research, register and become researchers. We then got a quick tour of the researcher workbench where we looked at a few core support materials, and then we went on to do our own project, working with not only phenotypic data, but also genomic data within the researcher workbench. So with that, I'm going to conclude my portion of the demonstration and close, and I believe we have some time for questions.
Okay. Well, thank you so much, all three of you. Before we get into general discussion, I'd like you, I know Jeff and Sherry, you've been perusing the chat box and making sure that some of the questions have been answered. Just take another look at them again, see if anything fell between the cracks. So just to lead off the discussion here, and this is really a treasure trove of a data set that I can tell you members of our office here at CDC have been plowing through and trying to learn the processes of engagement and so on and so forth. But other than the steep learning curve, once you go over the initial hump, things will get easier. Just want to lead off the discussion by sort of comparing and contrasting, and maybe asking Jeff and Sherry a little bit about the sampling frame for all of us, because we in public health and doing population studies are accustomed more or less to population-based or representative samples like the National Health and the Examination Survey, NHANES, or NHIS, the National Health Interview Surveys. These are data sets that allow you to generalize prevalence of different diseases or risk factors or associations. All of us is different. I mean, it's a volunteer army of participants that come in. And I'd like to ask that question because we've been struggling with it. We've talked about it, and I would like to see how we can move forward with the idea of using this rich and large data set beyond the initial sort of inclusion of underrepresented populations in biomedical research to make it truly and all of us for the US at least. I mean, how can we go from 1 million people to 300 million people and try to get the best out of the sampling frame of all of us to make it more, quote, representative of the population? And that's sort of the elephant in the room. I've had many discussions with public health researchers who express a certain degree of skepticism about the representativeness of all of us. But I think these things can be worked through. And I would like to hear a little bit from you, Sherry and Jeff, about the sampling frame for all of us and how we can make it truly representative, both analytically and further in recruitment, let's say for the next 500,000 people that would come in. So any comments along those lines would be appreciated.
Sure. So first, I'll start off and say that I don't think the All of Us research program was necessarily intended to be a national study of the type that you're talking about. You may have seen that Sherry presented data on nearly 600 million novel genetic variants that were seen in the first 100,000 genomes, attributable, I believe, to the fact that we have individuals who have never been seen in biomedical research databases in the past using genomic data. So we have to recognize the power of the All of Us data set to unearth and unmask important genetic as well as phenotypic information, social determinants of health. There are other things beyond the genetics, of course, that we're going to learn about our population that we've never known before. So the balancing act is to strive for what you're looking for, Muin, in terms of a national representative cohort, and also to be able to really bring the diverse population data forward. So as you know, we've discussed this between our teams, and we're hoping that that with astute epidemiologic methodologic approaches, we can begin to at least potentially mathematically or otherwise be able to impute the representative data that you're looking for. I'm not sure that we're prepared to change our recruitment strategy for the next 500,000. That's probably not something that will be easy to do or necessarily desirable. So, with 1 million plus individuals, the question for the epidemiologists in the room is how can we carve out a subset of our cohort, perhaps, that is truly representing the U.S. population. I don't know, Sherry, if you have other comments along those lines.
Nothing more to add, except we do hope to bring in those weights, as you mentioned. So
Good.
It is something we're thinking very carefully about.
Okay, thanks. So for those people in public health who are not just interested in the genomic data like I am and our office at CDC, would like you to all think about the use of all of us in many, many domains. I mean, you heard the presentation of the type 2 diabetes. There's so many questions that can be asked. One question I have for Sherry about the ancillary studies, because, with a platform like this where you're doing slow, meticulous work, collect data over time, how do you go after the low-hanging fruits in terms of adding more ancillary studies to this, and how can you show early impact? Because my biggest comparison to all of us is the UK Biobank, which has been around for decades, and it took years for the data to come in, and now we see the publications. But what is the approach for all of us to early wins, early impact, and adding ancillary studies, if I could ask?
Yeah, we're really trying to highlight some of our early wins from the whole cohort overall. Ancillary studies were something that we're just starting to stand up as part of the All of Us research program. So, for instance, we launched the Nutrition for Precision Health ancillary study this past year, which is really diving in deep on nutrition and health. And we will be ramping up the amount of ancillary studies that we have and the amount of data from those ancillary studies that feed back into the researcher workbench. So I don't know that early wins are really necessarily from ancillary studies, but we have a lot of early wins from the whole cohort, and we'll continue to hopefully have really amazing breakthroughs from both the whole cohort as well as the ancillary studies.
So along those lines, Sherry, if you would speak a little bit, because at the start of the pandemic, the initial cohort of people and all of us were already recruited, and they had data that seemed to have been very useful in publishing early results about who's been infected and so on and so forth. Could you comment on some of the early COVID work was done in all of us?
Sure. Yeah. Once the pandemic was rearing its ugly head in March of 2020, we realized that we were really well positioned to look at when the pandemic actually started in the United States. And so we actually did a serology study on our stored serum with Quest Diagnostics in a CLIA certified environment. And we did a pretty rigorous approach using some help from scientists at CDC as well as other places, where we did the test on both the spike protein as well as the nuclear protein to make sure that the person really had SARS-CoV-2 and not another SARS virus. And actually returned those results to participants, which gave them an idea of like, we got some responses like, God, I knew I had it earlier. And, there was no test around. There was nothing to tell me that I had it. So we were actually able to show that the first cases started likely in December of 2019, using the valuable resource that we had. And so hopefully we can continue to do things like that as needed. Hopefully there's not another pandemic. But we really hope that we can pivot and use our valuable resource for studies such as that.
Thanks, Sherry. Looking at the
Hey, Moen, can I just jump in for a
Yes.
second?
Go ahead, Jeff.
Just to say that to your earlier point about low-hanging fruit and ancillary studies, I think there are two things to recognize. I would say the unprecedented nature of having the 5 major data streams that we presented today: EHR data, biometric data, genomic data, wearable data, and survey data in one place on most of the participants is something where we would hope that novel insights could be achieved both in terms of disease biology as well as translatable findings for impacting population health. So to Sherry's point, we should not need more data to really optimize or maximize the value of what we've already done. And having said that, the biospecimen collection is enormous. And so the next probably earliest ancillary studies that we'll do, like the serology study that Sherry just mentioned, will be translating those biospecimens into data, whether it's other omics, RNA profiling, proteomics, metabolomics, immunophenotyping. I think we're in the process of developing a scientific roadmap that will dictate the first uses of the biospecimens over the next 12 to 18 months. So I guess we'll stay tuned for that, but I encourage people not to wait for that and go at the data that we already have.
Thanks, Jeff. Very helpful. Perusing the chat box here, and I see that questions are being answered in real time. So thank you all, and for doing that as we go forward. To me, some of the greatest assets of all of us is this the wealth of data and the longitudinal approach to data as we all try to develop more predictive analytics for different diseases, the course of lab measurements over time. I think that this would be a rich area for what I would call precision public health, try to sort of move the needle in terms of prediction for individuals and populations. But it's complicated. It's not going to happen overnight. And I would like to ask the people who are on this presentation today, the participants, we have 133 people still left over, as to think with us about the kinds of public health questions and that this data set is uniquely positioned to answer for your programs within CDC or state public health, whether it's diabetes, as you heard, or COVID-19. This data set has different assets that would allow sort of a unique look at some of the public health topics of importance. So think with us about this, and don't be shy about reaching out to our office and also to the rich resources that the data center and the help that they provide in getting started. So this is really an amazing feat. And I'm looking at the clock, it's 10:30 here, but I don't want to shortchange. We have another half an hour if people want to continue the conversation. So one more, it's coming in. I don't know if you want to answer those online, but I'm open to people asking more questions if they want to. This is the beginning of a conversation that we have between the world of public health and the world of biomedical research at NIH. And we're really, I mean, I'm personally thankful for this kind of a unique data set that would allow these kinds of questions to be asked and hopefully refined over time. And so any questions, any last questions or thoughts from anybody? I'm giving another minute here. Browsable list of all data elements publicly available online somewhere. Yes, the answer is yes. If you want to point in that direction, Kelsey.
Sure. And I can answer the question about Jeff. I saw you had your hand raised.
Okay, you go first.
Are you okay if I-
Yeah. You don't need to raise your
so-
hand, Jeff. Just butt in. Just interrupt me anytime. Okay, go for it.
So I think there was a wonderful question about the ability to see, you know, how many diabetes or how many participants who have diabetes also have genomic data. So this really gets to the linkage between some of our phenotypic information and our genomic information. And I think that this is a wonderful area. The program is very interested in expanding our public tools to enable you to dig into these type of associations. Right now, you cannot do that, but this is something that we're very actively working on. So I hope that you will be able to do that in the not too distant future. So great question. I completely agree that would be valuable and we're working on it.
Jeff.
Yeah, and my comment really is to follow on to Muin's point about this community of researchers that's on this call today. You saw Kelsey present the 2,600 projects at the Researcher Workbench. And one way to view that is just a catalog of projects, but another way to look at it is an opportunity for collaboration. Since you can look up areas of interest to you or to your teams and identify researchers who are already potentially moving that agenda forward, reach out to them and use the Workbench as not just a place to analyze data, but to forge relationships and collaborations that will make it even more powerful.
Yeah, along those same lines, I mean, our office has been sort of tackling the issue of family health history because all of us data has multiple ways to ascertain family history. One from the survey, the other is from electronic health records. And then, this is a rich area for public health genetics that I invite anybody who's interested in any disease to work with us and the many groups that are tackling family history as a public health issue. And of course, once you start linking the family history with the genomic data, you'll be getting a lot of information and also the environmental variables. There was a question about rare diseases in there. I think it flashed very quickly. I think the size of the cohort can allow a little bit of a look on rare diseases. Certainly, it depends on how rare is. But any comments on rare diseases from any all three of you? Let's see, where's that comment about rare? Are there any efforts to characterize rare diseases in this data set?
I think the answer is that it's of great interest. It's one of the areas of opportunity for identifying variants that are actionable that lead to treatments and potential cures. But it has not been a major focus yet. And we would encourage participants who have rare diseases to join the cohort so we can learn more about them and also to utilize the the vast array of novel variants to help inform rare disease diagnosis. And the last thing I might say about this is that we're hoping that one of the things that will come out of this work, along with others around the world, is a full catalog of loss of function alleles across the human genome, which will be a very powerful tool for rare as well as common disease, we think.
Thank you, Jeff. Any other thoughts, questions here? Somebody direct messaged me about making the recording available. And so we want to thank Vanderbilt for doing that. Hopefully this whole hour would be online soon, and the slides will be available as well.
Kelsey, do you want to talk about cost? So there's a question about, I think.
Good question, yes.
Yeah.
Absolutely. So right now, there is no access fee. So you are not charged for accessing the data set. You are not charged for using tools like our cohort builder, et cetera. We do incur some cost for compute. And so when you are using your virtual machine to do, let's say, that genome-wide association study I showed earlier, you pay for compute. And to sort of offset this cost, particularly as you're learning the program and perhaps running a few tutorials, we do provide researchers with an initial set of free credits. And so this really, we find, is usually sufficient for a lot of exploration. And certainly if you're only working with survey data or EHR data, many projects can actually complete their entire studies using those free credits. As you use larger data types, as you use more powerful VMs and incur more compute cost, you will have to set up a billing account. We have a lot of resources on how to do that. And so I would direct you again to that user support hub where you can search for billing and see a lot more information on that. One thing just to note about cost is, while there is a cost associated with using a workbench, if you're using this virtual machine, it still is very affordable. And so the reason I say that is when you think about the fact that all of us is actually paying for the cost of storing this data, and we've taken some steps to optimize your work, I think that this really is still trying to lower barriers to accessing and analyzing this data. But do be mindful, if you're trying to ramp up a powerful machine to do a huge analysis, it will cost money.
So along those lines, as part of the interagency agreement between CDC and NIH, I'm here to tell you all, at least the ones, the scientists from CDC, is that you have nothing to fear. You can use the data without worrying about the cost, at least for the next 2 years, starting in this fiscal year, which is almost done. And next year, we set it up in a way that it will allow you, even if you exceed that initial allotment, not to worry about it. Now, what we'll do after that remains to be determined, and certainly that doesn't apply to the non-CDC scientists who are on this webinar today. Okay, social determinants of health. Sherry, you answered that one. Follow-up question about rare diseases. Anybody wants to tackle that one? By Xiao Fan. Okay, lots of questions, and any other thoughts? If not, I'm going to close up this meeting today and thank.
Maybe we can answer the Clinborg.
One question which is worth mentioning. I got asked, can you download the data? No.
No.
You cannot download the data. You must use the researcher workbench. You must do your analysis there. I feel like it is worth saying that, which is, I think, really, really important to understand. You can download aggregate results that meet our dissemination policy, but you cannot download the data set to work locally. Okay, sorry to end on that note. I think to bring it back up to a high level, I think, the reason that we do that is because we have hundreds of thousands of participants who have entrusted us with their data. So be mindful as you onboard into this incredible resource that we need to honor their contribution. We are determined to protect our participants because without them, we don't have this program. And so all of us takes this very seriously. So I think as you are all on the call and thinking about the ways that we can advance health, I think this program really underscores and thanks our participant partners. And we challenge you, drive forward the next generation of research that really improves population health for all of us here in the United States and beyond. So I'm so excited about seeing what this research community will do. Thank you so much.
I think this is a good way to end the meeting. So thanks to all of you for attending, and our three speakers today. We'll try to post this the webinar as soon as we can, and hopefully I'll see some of you next week to talk about COVID-19 and host genomics. So thanks all, and have a great rest of the day.
Thanks so much.